Hello, everyone. My name is Boris Aguilar. I'm also in Ilya's group. And the next speaker will be Reinhard Laubenbacher, who is a professor in the Department of Medicine, director of the Laboratory for Systems Medicine at the University of Florida. Ilya and Reinhard have known each other for many years, almost 20 years. They have very common interests in mathematical biology, on using Boolean networks. And recently, they both got interested in developing digital twins. They published some work together recently as 2024. And Reinhard will talk about digital twins. Thank you, Reinhard, for joining us.
Thank you very much. It's a real honor to be here. As Boris says, I became aware of Ilya's work about 20 years ago. I moved into mathematical biology. I was a theoretical mathematician. I would prove theorems about all sorts of things, and then pretty abruptly moved into biology. And so Boolean networks to me were a quite natural thing to look at, and we found Ilya's work on probabilistic Boolean networks. We've used them ever since. I've had many discussions with Ilya about them. But we've just heard a great introduction to Ilya's work. I don't want to talk about Boolean networks. I do want to talk about digital twins, which is new to everybody in this field. It's, you know, for maybe 2 or 3 years that this has been a concept, and it's taking off quite a bit. So let me. It's very confusing. Very few people know what it means. Maybe nobody knows what it really means or should mean. I just spent 3 days at a workshop at the NIH earlier this week, engaged in somewhat in the exegesis of a National Academy's report that came out last fall, and in trying to figure out what specifications and requirements for a digital twin project ought to be. And if you read the report, the message is much of what's out there is aspirational, and many foundational mathematical, computational, and data science problems need to be solved before this becomes a viable technology. So this is, it's quickly becoming the most cited figure in digital twin anything from that report. The report focuses not just on medical digital twins, but also on engineering, climate science. The foundational problems are all the same. We just heard a great introduction to what a digital twin should be that conforms pretty much to what this report outlines as being a digital twin. It's becoming pretty quickly the accepted definition. And whenever you go to any sort of digital twin meeting, a large and increasingly tiring part of the discussion becomes, what is it? In part, people don't know. In part, people intentionally misrepresent what they do. If you're interested in this, it's a great thing, but it's complicated, and it's going to be expensive. So as was said earlier, digital twins come from industry. In 2013, General Electric introduced a data analytics platform that focused on its own products, you know, turbines, wind turbines, jet engines, and so forth. And they would take streaming data off of these products and massage them to put into specifications of these pieces of equipment, and then use them for preventive maintenance and so on. And the way they explained it, just to build this data transfer and analytics tool, they spent roughly about $1 billion. That was 15 years ago. So it says something about how complicated these things will be, because every digital twin product is essentially a soup to nuts, bench to bedside project that results in a product to be sold in some form or another. So in February of last year, I organized a 2-day workshop on discussing issues around digital twins in specifically related to immunology. It was brought together 20 people, and the idea was that everybody would present for 15 minutes their thoughts, their projects, and then for each of these presentations, there would be a half-hour discussion with other discussions sprinkled in. So the entire 2 days were very much focused on discussions. The only audiovisual tool was a whiteboard, so no PowerPoint presentation. So it really was focused on on discussion. Of course, I wanted to invite Ilya and gave him the option of participating via Zoom because I knew about his illness, but he decided to join and spend the 2 days there. We published a couple of basically reports from this meeting, and then independently, Ilya, my colleague Bonamerat, and Natalia Trajanova from Johns Hopkins published a review/perspective on this issue. It's part of a special issue on digital twins in Nature Computational Science, I believe, yes. So there Ilya also made a big contribution, and it was really before he got very sick, so I very much appreciate that. Now, I could spend some time describing to you the project that Ilya presented, or our work that's related to it, but I think it would be best if I let Ilya speak for himself. We recorded all these proceedings, and so I want to just play for you the first 15, 18 minutes of this, where Ilya explains his AML digital twin project. And so if you could play that, please.
Thank you so much again, Reinhard, for inviting me. What I'd like to do in my 15 minutes is to give you 2 sketches. They're very sketchy. The first one is showing how we think about the life cycle of a.
Sorry, one moment. Just trying to get the audio to go through.
Digital twin. And the second one will be more specific about AML digital twin for acute myeloid leukemia. As I mentioned when I introduced myself, we have started a project to develop a prototype for the AML digital twin. So I completely share the optimism of several in the room, Tom, others, that we can get somewhere. It will never be perfect, but if we can do a little bit better than standard of care, then that's already a great step forward. So first, I'd like to start with the digital twin life cycle. We have a patient, and we generate multi-scale and multimodal data from the patient. When the patient comes into the clinic, various kinds of data. It depends on the tumor type. For AML, it's going to be some specific data types: flow cytometry, cytogenetics, mutation panels, so on. Clinical data, of course. Then we use this data for building models, training the models, or I should say calibrating the models to the data. And by the way, I should say that this figure that I'm reproducing is published in a commentary in Nature Medicine. Paul Macklin is a co-author, my partner in crime for this, and also several other collaborators in Stanford. The National Cancer Institute, Department of Energy, it's a group of us that conceptualized this view of the cancer patient digital twin. So the model is then used to explore digital twin predictions. So this could be disease trajectories, predictions of toxicity, adverse effects, responses to treatment. So it's a computational exploration. So these explorations have to be visualized. This is my terrible attempt to draw a laptop. This is a keyboard. It has to be visualized with some user interface in what we're calling a dashboard. So these predictions can be interactively explored by the doctor and the patient. So here's the doctor, here's the patient. They are working together. The patient is participating in the decision-making to the extent that the patient wants to participate. Many patients feel like they just receive the recommendations, but they don't feel that this is a participatory process. And the digital twin will help enable this aspect of clinical care. And then individual care decisions will be made. Individual, emphasizing that every patient is unique at some level, and every decision must be tailored to that individual. Of course, people who actually do practice medicine will say, well, it's already that way. We already try to individualize the decisions. We do the best we can. But this is going to be supported by these computational models, which are constantly updated. So there is a continuous learning cycle where, as many patients are studied and treated with digital twin technology, we will have then cohorts of digital twins. We will even be able to create cohorts, virtual cohorts of digital twins. And with such capabilities, we will be able to do population studies. We will be able to advance basic science. So for developing new interventions, for example, understanding the mechanisms better. And we can even do virtual clinical trials, or enrich cohorts for actual clinical trials. So there is a continuous learning loop. Continuous learning. As more and more patients are engaged in this, the digital twin technology will get better. But also, over multiple clinical encounters, even with one individual patient, the actual digital twin for that patient will also get better. It should get better because there's more data that is collected. So whatever calibration of the parameters is performed, it will be refined as more and more data are being used. So this can be used to adjust treatments It can also be used for monitoring. Hope you can read my handwriting. Monitoring response to interventions, to treatments, therapies, and even track lifestyle modifications. So for example, another project we're working on is diabetes digital twin. This is where lifestyle modifications are very, very pertinent and consequential. So this is the life cycle of the digital twin. I'm sorry, I'm going to now go to here, and maybe you won't, I'll try to keep it here so you can see it. Sorry, Reinhard and Luis, I know you're in that zone. So now, AML, acute myeloid leukemia. Obviously, it's related to the immune system. It's a blood cancer. It's a hematological malignancy. There are multiple types of leukemia: ALL, CML, other blood disorders, MPN, and so on. We decided to focus on AML because it has, not only because it has dismal prognosis, but also because patients routinely get multiple measurements over time, relatively non-invasively. They're bone marrow biopsies and aspirates, which are quite invasive and intensely painful, but they do get, multiple such biopsies throughout the course of the disease progression. But they also routinely get peripheral blood draws, and that is also quite informative of disease, with information like CBC differential and various other panels like metabolic, renal, other kinds of panels that are informative of not only disease, but also side effects of treatment. Because many patients, after they receive various chemo drugs or other interventions, they become, for example, cytopenic, pancytopenic, or neutropenic. So they don't have neutrophils, they become vulnerable to infection, they become thrombocytopenic, they don't have platelets, they don't have red blood cells, they can bleed, they need transfusions all the time. So patients get blood draws sometimes daily. So you can get data, very nice time series data, something you cannot get from solid tumors, right? From, if you imagine something like pancreatic cancer, brain cancer, it's very difficult to get multiple time point measurements unless it's imaging or maybe biopsies. But with blood disorders, you can get really good data. And so that's a good starting point for building a digital twin, because we have this time series data. So again, here's the patient and the doctor. The patient, there are various kinds of data types. Maybe this box is too small. So there's clinical data, obviously I mentioned, from peripheral blood, from bone marrows. There is flow cytometry. This is routinely collected in all clinics. I mean, the actual assays may be different, what markers they use for flow cytometry. But generally, it's going to be the same for identifying the percent of blasts, which are the leukemic, the cancer cells, but also other cell types in the bone marrow with different markers. Flow cytometry, cytogenetics. So looking at large-scale rearrangements and aberrations, genomic aberrations, which can be used to stratify patients into different risk groups. There is molecular data, such as genetics, as I mentioned, genetic panel. So these are mutation panels. These are also very informative as for deciding which drugs might be effective. Immune profiling. This was mentioned before in one of the presentations. Cytokine profiling. Now, some of these things are not routinely done, but they can be. Of course, there's a cost associated with it, and even transcriptomics, like RNA-seq from the bone marrow, or single-cell RNA-seq, which can show you the different populations of cells, including the leukemic cells. And then there are also treatment response measurements. After the patient is treated, there are various measurements that can be made to look at response. So here's my laptop picture again. There is a dashboard that is used by the doctor and the patient to visualize the predictions of the digital twin, visualize the historical data, like a dashboard, and this is actually used at the Fred Hutch. I'm working with collaborators in Helsinki University Hospital in this project, as well as with the Fred Hutch Cancer Center. And they're already using a dashboard to visualize retrospectively, the disease progression, when the patient was diagnosed, what treatment was on a timeline, when the treatment started, when it finished. You can overlay various, side effects, anything, cytopenia, shortness of breath, liver function, and so on. You can see historically what was going on. But right now, you cannot look into the future. But with the digital twin technology, we will be able to use the dashboard to also look into the future, make predictions, predict drug response, for example. And we will be able to predict adverse events. As I mentioned, things like cytopenia, for example, which is very important. The actual engine of the digital twin is going to involve ensemble type of modeling. Not just one model, many types of models, depending on what it is that we want to predict. It will include multicellular modeling. This will be typically agent-based modeling that will explicitly model stem cells, hematopoietic stem cells, leukemic stem cells, progenitor cells, other cells such as lymphocytes, particularly in the context of allogeneic stem cell transplantation. This becomes very relevant because the immune system can exert a graft versus leukemia effect. So the T cells can actually kill the cancer cells. Of course, they can also cause graft versus host disease. So it's two sides of the same coin. But this multi-cell model will try to capture these effects with measurements, obviously based on measurements and tuned to these measurements. This model, there will also be a genetic regulatory network model, GRN, which will model on the molecular level the aberrations of genetic regulatory networks in AML cells, in leukemic blasts. And these models will be used to generate features. And then these features will be input into a machine learning phase of the model. So you have features from multi-cell modeling, from genetic regulatory network modeling, and there may be multiple models even within these boxes, but they will all be used to generate features. And this will result in personalized models. These models, the machine learning, will be used to visualize these predictions in the dashboard. There is a lot of public data. AML is also quite a good cancer to study because there are many large data sets for functional screening, drug screening, genomic data sets, cell line data sets. I'm not going to list all of them, but there are quite many. So from these public data sets, I'm just going to draw this kind of a database. So you have drug screens, functional screening, you know, things like with CRISPR screening, multi-omics data sets. There's, for example, a big data set called Beat AML, which does genomic transcriptomic profiling together with drug screening, ex vivo. So they get the primary patient cells, do the screening ex vivo, have the results, you have the data. Also, of course, clinical trials. And so all of these data can be used to generate so-called knowledge graphs. Knowledge graphs. These knowledge graphs, and we've already done a lot of this work in my lab, so they're based on general knowledge graphs that are not disease specific, but once you start overlaying these data on the general knowledge graphs, they become disease specific knowledge graphs, essentially AML knowledge graphs. These knowledge graphs can be then used to engineer features that also go into the machine learning stage. They also can be used for creating the gene regulatory network models. So this machine learning box essentially integrates features from all of these models, mechanistic models, network models, knowledge graphs, to make individualized predictions. I should have also drawn an arrow here that shows model integration. I'm finished. You didn't have to say, just got up and that's enough. Yes, we're well trained. So all these data are of course used to instantiate the models and to calibrate the models as I showed over here. So this is a 5-year project. We just started this year on this project, but we've done a lot of preliminary work on drug target prediction using these approaches. We're now starting to work on mechanistic models to predict disease progression, even in the absence of therapy. Okay, I'm going to stop there and we can have a discussion. Thank you.
Okay. So I encourage you to watch the rest of this. There's another half hour. And I also encourage you to watch the rest of the other session recordings, because Ilya was very active in participating in the discussions around a number of projects. And as you can imagine, over a 2-day period, when you mostly just talk, you really get to learn about what people think and how they view different aspects. So from that point of view, this was an incredibly instructive and interesting event. Now, during it and afterwards, I talked to Ilya quite a bit about his project, and in particular, his plans on actually turning this into a commercial product. He was setting up a company. He had hired a CEO, and we were talking about ways that we could maybe work together with them. Now, of course, tragically, Ilya was not able to benefit from this research himself, but I'm sure that this will carry on, and many people in the future who suffer from this disease will. So, you know, those of you who are colleagues of Ilya, I'm sure, have more than once heard some version of what he just did here. But I think for the rest of us, this was just a wonderful introduction. As Liud said earlier, Ilya was a great teacher, and it clearly comes through in the way he described this project. So thank you.